Did you know that roughly 59% of the world's biodiversity actually lives in the soil?
Yeah, it's a wild statistic.
Right. Not the rainforest canopy, not the coral reefs, but literally directly beneath your shoes. So, welcome to our deep dive. I am so thrilled you're joining us for this because today we are taking a huge stack of, encyclopedic excerpts, research notes, and biological surveys all about soil biology and the soil food web.
And we're going to explore exactly how this completely hidden ecosystem cycles nutrients to support basically every piece of terrestrial plant life on Earth.
Exactly. And it's a critical mission for us today because, let's be real, if you were to somehow just remove this microscopic food web from the equation.
The entire terrestrial ecosystem would just grind to an absolute halt.
Yeah.
Every single fallen leaf, every dead branch, every piece of organic waste would just pile up indefinitely. I mean, we would literally be buried under undecayed litter. There'd be no humus creation, no nutrients returning to the Earth, and ultimately no plant life at all.
Which really puts the stakes into perspective right away. We're looking at the ultimate, recycling mechanism. And the sources we're diving into today introduce this collective term for all the life driving this mechanism, which is the Edaphon.
Right, the Edaphon.
Yeah, and this encompasses all the living organisms, the biota and the fauna, that spend a significant chunk of their life cycle down in the soil profile itself, or right at that boundary layer where the mineral dirt meets the leaf litter on the surface.
And when we talk about the Edaphon, you have to conceptualize a population scale that is, honestly, almost impossible to grasp visually.
Oh, totally.
So scientists categorize these organisms into four main groups based entirely on their physical size, because, size pretty much determines exactly how they interact with the physical architecture of the dirt.
Right, so the scale ranges from what we know down to the totally invisible. At the very top, you've got the megafauna. These are organisms 20 millimeters and larger.
The big guys.
Yeah, exactly. We're talking about vertebrate burrowers like moles, rabbits, gophers, prairie dogs. And then if you step down a level, you have the macrofauna.
Ranging from 2 to 20 millimeters generally.
Yeah, these are the creatures you might actually see if you go out and turn over a rock in your garden. Earthworms, woodlice, beetles, millipedes, slugs, ants.
And that boundary right there is roughly the limit of what we humans easily notice on a day-to-day basis.
Right.
But the really vital work of the Edaphon extends way, way below that threshold. Under the macrofauna, you have the mesofauna.
Okay.
These range from 100 micrometers up to 2 millimeters. This is the domain of, tardigrades, mites, springtails, and those tiny enchytrate worms.
And finally, the foundational layer, right? The microfauna and microflora.
Yes. Measuring between 1 and 100 micrometers. So we're looking at yeasts, bacteria, archaea, fungi, protozoa, and nematodes.
And what I think is so essential to understand about these four really distinct size categories is that they absolutely do not operate independently.
Oh, not at all.
They don't just happen to share the same patch of dirt. They're bound together in these incredibly complex trophic networks, which, we just commonly call the soil food web.
Right. And I actually really like the analogy of a city for this.
Oh, right. The metropolis idea. Let's unpack that.
Yeah, because instead of thinking of soil as just like a dumb sponge that holds water for plant roots, you really have to view it more like a highly stratified metropolis. Every citizen has a very highly specific job in waste management or chemical refinement or resource distribution.
I love that because it emphasizes that plant health is a really active, ongoing process. A plant isn't just sitting there passively soaking up whatever happens to float near its roots.
No, it relies entirely on the continuous labor of this city to survive.
And that labor basically starts with breaking down detritus. Like when a leaf falls off a tree and hits the ground, it's essentially locked up, right?
Exactly. The plant that dropped it cannot reuse the carbon or the nitrogen or phosphorus inside that dead leaf.
So the primary decomposers, the bacteria, the archaea, the fungi, they have to physically dismantle that organic material. They break it down to produce detritus.
And from there, saprotrophs, which are organisms that feed on decaying matter, they come in and extract the soluble nutrients from that newly formed detritus.
And this chemical extraction happens most intensely in a really specific zone, right?
Yeah, a zone called the rhizosphere.
Yeah.
Which is this incredibly narrow, biologically explosive region of soil right around the plant's roots, completely influenced by the root secretions.
And what I found so fascinating in the sources about this process is this concept of biological lockup.
Oh, yeah. That's a huge point.
By eating the detritus and pulling the soluble nutrients straight into their own microscopic bodies, These organisms are actually preventing a massive environmental failure.
Right, because those nutrients were just floating loose in inert sand or clay.
The first heavy rainstorm would just wash them completely away.
Exactly. They'd leach down into the deep subsoil or just wash out into the groundwater, totally out of reach of the plant roots.
So the Edaphan basically acts as a living reservoir. The nutrients, especially those highly mobile elements like nitrogen and phosphorus, they're held safely inside the biomass of the soil life itself.
And they're only released back into the soil in plant-available forms when those organisms, excrete waste, or when they die and get eaten by another member of the food web.
Which cycles the nutrients locally, keeping them precisely where the roots actually need them.
Precisely.
So, to really understand how this metropolis manufactures and holds onto these nutrients, I think we have to start at the bottom tier.
We need to look at the workforce doing the primary chemical heavy lifting, the bacteria.
Yeah, and the sheer volume of the bacterial workforce just totally defies our normal sense of space.
It really does. I mean, bacteria are single-celled organisms, right?
Yeah.
And they are the most numerous inhabitants of literally any productive ecosystem.
The sources put this into a perspective that requires you to really pause and think. If you go out to a healthy agricultural field or just a rich garden bed and you scoop up a single teaspoon of soil,
Just one teaspoon.
yeah, you are holding a population ranging from 100 million to 3 billion individual bacteria in one teaspoon.
It's mind-blowing. And their power comes not just from that standing population, but from their biological capacity for exponential growth.
Right, because bacteria reproduce through binary fission.
Exactly. A single cell elongates,
Yeah.
replicates its genetic material, and divides into two identical cells.
And the speed of that division is staggering. The text points out that under favorable conditions, an organism like E. coli can enter this exponential phase of growth where it is capable of producing 1 milliard, which is a billion more cells, in just a single hour.
Wait, 1 billion cells in an hour.
A billion in 1 hour.
That is just incredible. And when you have an almost infinite, rapidly replenishing workforce like that, you get an ecosystem that is highly, highly reactive to its environment.
Which makes sense.
Most of the beneficial bacteria that drive plant health, they congregate right up against the roots in that rhizosphere. And these rhizobacteria, they live entirely in the soil water.
Which, just to clarify for everyone listening, that doesn't mean puddles or flooded ground, right?
Oh, no.
When the sources talk about soil water, they're referring to this microscopic, incredibly thin film of moisture that just clings to the surface of individual dirt particles.
Right. And some of these bacteria actually use tiny whip-like tails called flagella to physically swim through this microscopic water film to seek out nutrients.
That mobility is essential, but it's also entirely dependent on the physical conditions of the soil.
Absolutely. Because the vast majority of the bacteria that benefit agricultural growth are aerobic.
Meaning they fundamentally require oxygen?
Exactly. They thrive when the soil is moist enough to provide that water film, but aerated enough to allow oxygen to actually penetrate the soil pores.
So if the soil becomes super compacted or just completely waterlogged after a heavy flood, that oxygen gets totally choked off.
And when the oxygen is depleted, the aerobic bacteria cannot function at all. So the anaerobic bacteria, the ones that operate without oxygen, they kind of take over the primary processes.
And the sources mention that while anaerobes obviously have their place in global ecosystems in an agricultural context, a dominance of anaerobic activity is not great.
No, it tends to result in the putrefaction of organic matter rather than, healthy nutrient cycling decomposition.
Right, which actually brings up a very practical question about the resilience of this microscopic workforce.
Oh, it was that.
Well, if you have billions of bacteria that absolutely require this perfect Goldilocks zone of moisture, oxygen, neutral pH, and food to thrive, what happens during a drought or a deep freeze? If the soil completely dries out, do these billions of bacteria just die off and leave the plants to starve when the rain finally comes back?
It's a great question, but no, they do not simply die off. The evolutionary pressure of living in the soil has resulted in these really remarkable survival mechanisms.
So what do they do?
When environmental conditions degrade, whether it's extreme heat, freezing temps, severe drought, the bacteria basically halt their growth phases and enter highly resilient dormant states.
They basically hit the biological pause button.
Correct. And the specific method depends on the type of bacteria. Certain gram-positive bacteria have the ability to form incredibly hardy spores.
Like little escape pods.
Yeah, these spores encapsulate the essential genetic material and can survive extreme environmental stress for really long periods.
Wow.
Gram-negative bacteria, on the other hand, might enter what is termed a non-culturable resting stage.
Mm.
Other bacterial communities will retreat into microscopic crevices coated in clay to form quiescent colonies, effectively hiding from the hostile conditions until the environment gets better.
The text also highlights something really intriguing about how these hostile conditions actually drive the evolution of the soil web.
Oh, the genetic adaptation part.
Yeah, it mentions that individuals with pre-adaptive mutations, or those that can rapidly evolve better traits, are the ones that survive the stress and outcompete others when conditions improve.
Right.
And this rapid genetic adaptation is heavily, heavily assisted by viruses.
The role of viruses in the soil is a rapidly expanding field of study, actually. The bacteria are constantly colonized by these persistent viral agents known as bacteriophages.
And it's funny because when most of us hear the word virus, we automatically think of disease, right?
Sure, yeah.
But in the soil metropolis, these phages are acting almost like biological couriers.
Exactly. They replicate inside the bacterial host, but as they move between hosts, they actively promote gene transfer.
They literally pick up pieces of genetic code from one bacterium and physically insert it into another one.
It's a natural mechanism of genetic swapping. It allows the bacterial population to rapidly share advantageous traits across the community.
Which is just wild that continuous open source genetic exchange is part of what makes the bacterial workforce so incredibly efficient at their primary job.
Yes. And from an agricultural perspective, the most vital labor they perform revolves around the nitrogen cycle.
Okay, let's really dig into the mechanics of the nitrogen cycle here, because this is where the bacteria function as specialized chemical refineries.
Right. Plants absolutely need nitrogen. It's a fundamental building block for amino acids and proteins.
Without it, a plant simply cannot grow leaves or stems. But the catch is, plants are notoriously bad at getting nitrogen on their own.
They really are. They rely heavily on three specific bacterial processes: nitrification, nitrogen fixation, and denitrification.
Okay, so let's start with nitrification. This is a transformative process that deals with the waste products of decomposition, right?
Exactly. When organic matter, like a dead insect or a fallen leaf, begins to decay, the proteins break down and release nitrogen into the soil in the form of ammonium.
And while ammonium obviously contains nitrogen, the sources note that many plants struggle to absorb and utilize it efficiently. It's essentially an unrefined raw material.
This is where the autotrophic nitrifying bacteria step in.
Huh?
Organisms like Nitrosomonas. They don't rely on photosynthesis or consuming organic matter for their energy.
So what do they eat?
They actually fuel themselves by oxidizing inorganic compounds. They take that unrefined ammonium and biochemically transform it into nitrites. And subsequently, other bacteria transform those nitrites into nitrates.
And nitrates are the highly refined, water-soluble premium fertilizer that growing plants can immediately absorb through their roots.
Right, they use it to build new plant proteins.
So they're literally taking toxic waste and refining it into high-grade food.
Exactly. That handles the nitrogen that is already locked in the organic cycle. But the total amount of nitrogen in a closed ecosystem would slowly deplete without a way to add new material.
Which brings us to nitrogen fixation. And this process feels like it borders on alchemy, let's be honest.
It really does.
Because the air we breathe is roughly 78% nitrogen gas. But that atmospheric nitrogen is essentially useless to a plant.
Right. The molecules are bound together so tightly that a plant root just can't break them apart. The nitrogen has to be fixed, meaning it has to be chemically converted into a usable organic compound.
And free-living nitrogen-fixing bacteria called diazotrophs, they actually possess the specific enzyme nitrogenase required to break that molecular bond.
Yeah, you have free-living varieties in the soil, but the most dramatic examples are the bacteria that form intense symbiotic relationships with the plants themselves.
The sources specifically highlight Rhizobia bacteria, which form a symbiosis with legumes like peas, beans, and clovers.
The mechanics of this relationship are just fascinating. The plant root actively recognizes the presence of the rhizobia in the soil and allows the bacteria to basically infect its root hairs.
And then the plant actually constructs specialized physical structures, tiny little nodules directly on its roots to house the bacteria.
It builds a custom refinery right on the supply line.
Yeah. The plant pumps carbohydrates down into the nodule to feed the bacteria. And in exchange, the bacteria pull that invisible, useless nitrogen gas straight out of the microscopic air pockets in the soil.
They crack the molecular bonds, synthesize it into organic nitrogen compounds, and hand it directly to the host plant.
Wait, so they just pull it from the air?
Literally out of thin air.
Yeah, that is wild. They are manufacturing fertilizer out of thin air.
It's difficult to overstate how critical this process is. Nitrogen fixers play a foundational role in primary succession.
Like when a new environment forms.
Yeah, they can enter a completely barren, newly formed environment, like rock scoured by a retreating glacier, and begin injecting the foundational building blocks of life into that sterile ground.
Wow. So we have nitrification refining the waste and fixation pulling new material from the sky. The third process is denitrification.
Right, and this process effectively runs the machinery in reverse.
While fixation adds nitrogen to the soil bank, denitrification represents a withdrawal. It's a series of processes that returns nitrogen from the soil matrix back into the atmosphere.
The bacteria responsible for this are typically anaerobes or facultative anaerobes, meaning they can switch their biological processes based on the presence or absence of oxygen.
Exactly. So when the soil loses its oxygen, again, think of severe compaction or prolonged flooding, these bacteria still need a terminal electron acceptor for their respiration.
And in the absence of oxygen, they switch to using the valuable nitrates in the soil.
They strip the oxygen from the nitrate molecules, converting the nitrogen back into a gaseous state like nitrous oxide, nitric oxide, or pure nitrogen gas, which simply floats away into the atmosphere.
The fertilizer essentially just evaporates.
And the text makes a really crucial point about human interaction here. It notes that an excess of artificial synthetic nitrogen fertilizers can actually trigger a massive denitrification event.
Oh, absolutely.
If you dump more soluble nitrogen onto a field than the plants can immediately uptake, and then the soil gets wet and loses oxygen, the anaerobic bacteria will just convert that expensive fertilizer right back into atmospheric gas.
Or it'll leach down into the groundwater, entirely lost to the crop. It really illustrates the delicate, continuous loop of the cycle.
Yeah.
A single atom of fixed nitrogen might circulate dozens of times, from a bacterium to a plant to an insect back to a bacterium, before a denitrifying process finally returns it to the sky.
So before we move away from a microscopic workforce, there's one highly specialized group of bacteria mentioned in the sources that we definitely have to talk about. The Actinomycetoda.
Ah, yes. Also known as actinobacteria or actinomycetes. They occupy a very specific niche in the soil metropolis.
They're basically the heavy-duty demolition crews.
Right.
While regular bacteria handle the simple sugars and easily degraded proteins, the Actinomycetoda are equipped with the specialized enzymes needed to break down the most stubborn, resilient organic materials on Earth.
They dismantle the tough cellulose and woody lignin from dead trees, and they even break down chitin.
Which is crazy. Chitin is a highly complex polymer, right? It's the primary structural component in the hard exoskeletons of insects, spiders, crustaceans.
Exactly. When a beetle dies in the soil, its shell doesn't easily decompose.
But the Actinomycetoda are uniquely capable of chemically dismantling that armor and returning those locked nutrients back into the humification process.
And they are responsible for one of my absolute favorite sensory details of the natural world.
Oh, the smell.
Yes. The sources explain that as the Actinomycetoda break down these tough materials, they produce specific volatile metabolites.
And those chemical byproducts are what create that rich, sweet, earthy smell you experience when you dig your hands into healthy, dark topsoil.
You are quite literally smelling the chemical exhaust of bacteria tearing apart insect shells and wood.
I love that.
It serves as an excellent biological indicator, too. If soil smells sour or metallic, the anaerobic bacteria might be dominating. But if you smell that rich, earthy aroma, you know the Actinomycetoda are actively maintaining the humification process.
And they're notably robust, too, right?
Very. While they prefer well-aerated environments with a neutral pH, they are significantly more tolerant of severe dry conditions than most other bacteria, allowing them to continue their demolition work even when moisture levels drop.
So we have these brilliant, localized chemical refineries processing the dirt, but there is a massive logistical problem here.
There is.
If bacteria are largely restricted to swimming around in microscopic water films, processing nutrients right where they sit, how does a mature oak tree, or even just a large tomato plant, get access to those nutrients?
Right, because a plant's roots are incredibly thick compared to a bacterium.
It can't possibly branch out fine enough to tap into every single microscopic puddle in the soil profile.
They physically cannot. The roots require an intermediary, a transport system that bridges the gap between the localized bacterial refineries and the plant itself.
Which brings us to the fungal kingdom. And the physical scale of the fungal network in the soil is almost hallucinatory.
It really is.
The sources state that a single gram of typical garden soil can contain around 1 million individual fungi, like yeasts and molds. But far more importantly, that single gram can hold up to 700 kilometers of fungal hyphae.
700 kilometers of physical structure woven into a pinch of soil you could hold between your thumb and forefinger.
I really had to think about the geometry of that. Wait, how is that even geometrically possible?
Well, to fit 700 kilometers of anything into a gram of space, those threads have to be unimaginably thin.
They're threading their way through the absolute smallest microscopic pores and crevices between individual grains of sand and clay.
Exactly, creating a massive three-dimensional web called a mycelium. They are exploring the microgeography of the soil to feed themselves.
Because unlike plants, fungi possess no chlorophyll.
Right, they cannot use sunlight and carbon dioxide to manufacture their own food. The biological term used in the text is chemoheterotrophic.
Meaning they require a chemical source of energy, much like animals do. They have to find organic substrates in the environment and consume them to extract the carbon and energy they need to survive.
And while the fungal kingdom certainly includes parasitic species that attack living plants, the vast majority of the fungi driving the soil food web are saprotrophic.
They rely on dead and decaying organic matter.
Yes. They actively participate in the degradation of freshly deposited biological remains, transforming them into humus and mineralizing the nutrients.
And the sources outline a distinct division of labor here, almost like a succession of specialized workers.
Right. When a piece of organic matter drops to the forest floor, the first wave of fungi to colonize it are those equipped with the enzymes to consume the easy calories, the simple sugars and starches.
Then, once those easily accessible carbohydrates are exhausted, that first wave of fungi dies back, and a second wave moves in.
And this secondary group possesses the much more complex enzymatic tools required to digest the tougher structural compounds, like the cellulose and hemicellulose.
And they digest the food externally, right?
Yeah, they exude the enzymes out of their threads into the soil, dissolve the material, and then absorb the liquid nutrients back into the mycelium.
And as they eat, they expand, sending their hyphae further out into the environment.
And the ultimate goal of that massive underground expansion is reproduction. When the environmental conditions are right, the sprawling hidden mycelium will push fruiting bodies up above the surface of the soil.
These are the familiar mushrooms, toadstools, puffballs. The visible mushroom is just a temporary, tiny appendage of a massive subterranean organism.
And its only job is to release spores into the environment to colonize new territory. Millions upon millions of spores.
They rely on the wind, they wash away in streams, but they also rely heavily on the broader food web.
Oh, totally. The text details how a massive variety of animals assist in fungal dispersal.
Everything from microscopic springtails to giant mammals. Like
Yeah.
a wild boar might dig up and consume a fungal fruiting body like a truffle, right?
And then walk several miles across the forest before excreting the spores in its droppings.
Effectively planting the fungal network in an entirely new region. That's amazing.
The physical expansion of fungi is remarkable, but their true power within the soil metropolis lies in how they interact with living plants.
Yes, we need to examine the mechanics of mycorrhiza. The word itself tells the story. Myco means fungus, and rhiza means root.
It is a highly evolved mutualistic symbiosis between a plant and a fungus. It basically operates like an underground logistics and trade network.
The plant roots are actively invaded by the fungal mycelium. The fungus lives a dual existence. Its sprawling network extends far out into the surrounding soil profile, while a portion of its structure resides directly inside the physical tissues of the plant root.
And the sources describe a few different architectures for this invasion.
Right.
You have ectomycorrhiza, where the fungus weaves a dense sheath completely covering the outside of the root and penetrates the outer cortex without actually entering the individual plant cells.
Forming a structure known as the Hartig net.
Exactly. Alternatively, you have arbuscular mycorrhizae, where the fungal threads actually breach the cell walls of the plant root cortex.
They form intricate microscopic branching structures inside the plant cells called arbuscules. It sounds like a parasitic infection, honestly, but it is a strictly negotiated trade agreement.
It really is. The fungus, lacking chlorophyll, desperately needs carbohydrates. The plant, bathed in sunlight, uses photosynthesis to manufacture those sugary carbohydrates in its leaves.
It pushes those sugars down its vascular system into the roots and willingly hands them over to the fungus.
So what is the plant buying with those sugars?
It is buying access to the fungal transport network. A tree's root system is thick, woody, and relatively inefficient at exploring microscopic soil pores.
But those 700 kilometers of microscopic fungal threads can reach infinitely further, wrapping around distant soil particles to extract moisture and scavenging critical immobile nutrients.
Particularly phosphorus, right, that the plant roots could never reach on their own.
Ah.
The fungus gathers these resources and pumps them directly back through the network into the plant.
It's an extraordinary biological barter system. The fungus mines the minerals, the plant catches the sun, and they trade in the dark.
But it goes so much deeper than a simple one-to-one relationship between a single tree and a single fungus.
Yes, the sources detail the concept of common mycorrhizal networks, or CMNs.
And this is where our understanding of forest ecology shifts dramatically. The mycorrhizal fungi associated with a specific tree don't just stop at the edge of that tree's root zone.
They extend outward, and they fuse with the mycorrhizal networks of neighboring trees.
They wire the entire forest together into a single contiguous biological circuit. It's basically a massive underground internet, which is just wild to think about.
It really is. This interconnected mesh allows for the physical transfer of resources between distinct individual plants.
The classic example provided in the literature involves the survival of saplings in deep, old-growth forests.
Right. If an acorn drops to the floor of a dense, mature forest and manages to sprout, it immediately faces a severe energy crisis.
The canopy above it is so thick that almost no direct sunlight reaches the forest floor.
Exactly. The seedling cannot photosynthesize enough carbohydrates to fuel its own growth. In isolation, it would simply starve and die.
However, because the soil is heavily wired with a common mycorrhizal network, the young seedling's newly formed roots immediately link up with the existing fungal threads.
And the older towering trees that have full access to the canopy sunlight, they push excess carbohydrates down into the fungal network.
The fungus then shuttles those sugars, along with water and nutrients, directly to the shaded seedling.
The older trees are effectively subsidizing the survival of the younger generation through the fungal broker.
It forces you to completely abandon the idea of a forest as a collection of individual trees ruthlessly competing for light and water.
Yeah, it operates much more like a cooperative superorganism, sharing resources to ensure the stability of the entire system.
And the text notes that these mycorrhizal associations are not an anomaly. They are the standard operating procedure for the vast majority of plant life on Earth.
There are notable exceptions, of course. Plants in the Brassicaceae family, which includes cabbages, mustards, turnips, they generally do not form these fungal associations.
Okay.
Other families, like the Ericaceae heathers and bilberries, utilize a highly specialized form known as ericoid mycorrhizae to survive in highly acidic, nutrient-poor soils.
But by and large, if you see a healthy plant in the wild, its roots are intricately wired into a fungal network.
And as these fungi grow and expand, they physically change the structure of the earth itself. The sources introduce a protein produced by these fungi called glomalin.
I want to save the most mind-bending detail about glomalin for the very end of our deep dive, but we have to explain its mechanical function now.
Right. Glomalin is a glycoprotein produced abundantly by arbuscular mycorrhizal fungi on their hyphal threads.
As the hyphae grow through the soil and eventually die back, they leave this protein behind.
And it acts quite literally as a biological glue. It coats the microscopic particles of sand, silt, and clay and binds them together into larger clumps, which soil scientists call aggregates.
This is what gives healthy soil its structure, that crumbly, spongy texture that allows water to infiltrate and roots to penetrate.
Without this fungal glue, the soil would just be loose dust, highly susceptible to blowing away in the wind or washing away in the rain.
And furthermore, because it is so incredibly stable, glomalin accounts for a massive percentage of the sequestered carbon and nitrogen stored in the soil.
It perfectly illustrates how deeply the biological and the physical are intertwined. The living organisms don't just live in the soil, they manufacture its structural architecture.
Which naturally leads us to the next vital question. We have the bacteria running the chemical refineries and the fungi managing the logistics and structural binding.
But an environment this active requires constant physical maintenance.
Right. Who is moving the earth, opening the pores, and regulating the environment so the microscopic workforce can function?
This brings us to the invertebrate ecosystem engineers.
We are stepping back up the size scale to the mesofauna and macrofauna, organisms like earthworms, ants, and termites.
The term ecosystem engineers reflects their profound ability to alter the physical and chemical dynamics of the soil on a massive scale.
The earthworm is arguably the most famous resident of the Edifon, but the specific mechanics of their job description are fascinating. We all know they burrow, but they are essentially living, mobile composters.
An earthworm sustains itself by ingesting vast quantities of soil particles mixed with decaying organic matter.
Let's look at what actually happens inside the worm. As that raw mixture of dirt and dead leaves passes through the worm's digestive tract, it is subjected to intense grinding and a bath of highly active enzymes and gut bacteria.
The physical and chemical transformation is severe. The worm extracts the nutrients it needs to survive, but the waste it excretes, the earthworm casts, is biologically transformed.
When that material exits the worm, the availability of critical plant nutrients within it, such as nitrogen, phosphorus, and potassium, is significantly higher than it was in the surrounding uningested soil.
They're continuously hoovering up low-grade dirt and manufacturing high-grade, highly accessible fertilizer.
But the chemical alteration is only half the job. The physical act of moving through the soil, what the scientific literature calls bioturbation, is equally critical to the survival of the metropolis.
Bioturbation is the biological stirring and mixing of the soil profile. As an earthworm pushes and eats its way through the earth, it creates extensive networks of physical tunnels.
And these tunnels act as the primary plumbing and HVAC system for the Edifon.
The sources point out that these burrows drastically improve soil aeration, allowing the vital oxygen to reach the aerobic bacteria deep underground.
Furthermore, they assure the ready infiltration of water, so when rain hits a compacted surface, it just
washes right off, causing erosion.
Right. But when it hits soil riddled with worm burrows, the water is channeled deeply down into the soil profile, exactly where the roots and the fungal networks can access it.
But perhaps the most elegant regulatory function the earthworms perform is chemical buffering. They actively manage the pH of their environment.
This mechanism blew me away. Let's explain why soil pH matters to the food web first.
The pH level of the soil dictates which microscopic organisms can thrive. As a general biological rule, fungal communities are highly competitive and active in alkaline soils, while bacterial communities dominate in more acidic soils.
So if the soil environment swings too far toward acidity, the bacterial refineries go into overdrive, but the fungal highways suffer and collapse.
And if it swings too far alkaline, the fungi thrive, but the bacteria lose their efficiency and the nitrogen cycle stalls.
For the metropolis to function optimally, both systems need to operate simultaneously,
which requires an environment that hovers right around neutrality. Earthworms, through their continuous ingestion and excretion of soil, act as a massive buffering mechanism.
They have specialized calciferous glands that secrete calcium carbonate into their digestive tracts.
When they excrete their casts, they're actively neutralizing the soil chemistry, pulling acidic soils up toward neutral and pulling alkaline soils down toward neutral.
It is an automatic biological negative feedback loop. They continuously stabilize the chemical environment to ensure the entire multi-tiered food web remains balanced and productive.
And they aren't the only invertebrates engineering the architecture. The text specifically highlights ants and termites.
Right. Ants and termites impact the soil structure differently than earthworms, primarily through their construction of massive centralized nests and mound systems.
The sources indicate that ant and termite activity physically concentrates the essential elements for plant growth, carbon, nitrogen, and phosphorus, in and around their colonies.
But their most impressive feat is the vertical mixing of the soil horizons.
In a static environment, soil forms in distinct layers or horizons. The organic rich material stays near the top, and the mineral dense material stays near the bottom.
But ants and termites are constantly running the freight elevators. A termite worker will travel deep underground, excavate mineral-rich soil particles from the subsoil, physically carry them up through the tunnels, and deposit them on the surface to build the mound.
In doing so, they are manually hauling trace minerals from the deep earth directly up into the active root zones, thoroughly mixing the layers and creating a much richer, more heterogeneous soil profile.
It is tireless, continuous manual labor that perfectly complements the chemical refinement of the bacteria and the logistical transport of the fungi.
They're constructing the physical framework that allows the microscopic world to thrive.
And that framework extends even larger. We've scaled up from microscopic bacteria through the fungal webs to the insects and worms. But the soil food web doesn't end at the surface.
It requires the interaction of large, above-ground animals to complete the cycle.
We must consider the impact of the megafauna, the vertebrate animals that utilize the soil.
We are talking about the heavy machinery. Mammals like gophers, moles, prairie dogs, badgers. They utilize the soil for shelter and foraging, but their impact on the hydrology of an ecosystem is profound.
When a prairie dog colony excavates a burrow system, they are displacing massive volumes of earth.
This deep burrowing creates immense nutrient-rich patches by aggressively mixing topsoil with deep subsoil. But their most critical function relates to water management.
The text notes that these large-scale excavations drastically increase the macroporosity of the soil. On a sloped landscape, heavy rainfall is a destructive force.
It rushes over the surface, stripping away the topsoil and carrying the nutrients into rivers.
But a landscape textured with vertebrate burrows captures that water. The tunnels act as massive drains, forcing the surface runoff to infiltrate deeply into the earth, preventing severe erosion.
And recharging the subterranean moisture levels that the bacterial workforce relies upon to survive the dry seasons.
And beyond the burrowers, we have the couriers, the animals that interact directly with the fungal network.
We mentioned the wild boars and the truffles earlier, but it is worth exploring how vital this is to the global survival of the forest.
Fungal spores are the seeds of the mycelial network. Above ground, mushrooms can rely on the wind to carry their spores across vast distances. But hypogeious fungi species that fruit entirely underground, like truffles, are trapped. They cannot use the wind.
So they evolved to hijack the olfactory systems of mammals. They produce incredibly potent aromatic compounds specifically designed to penetrate the soil and attract animals like mice, squirrels, and wild pigs.
The mammal digs up the highly nutritious fruiting body and consumes it. The millions of microscopic spores pass completely unharmed through the animal's highly acidic digestive tract.
The animal wanders across the landscape, perhaps traveling miles into new territory, before depositing those spores in a fresh pile of nutrient-rich fertilizer.
They are acting as the primary dispersal vector, effectively planting the fungal infrastructure for the forests of tomorrow.
It is an intricate web of dependencies. The broadcaster and naturalist David Attenborough actually coined a phrase to describe this specific dynamic. He called it the three-way harmonious trio.
The plant, the fungi, and the animal.
The plant captures the solar energy. The fungi mine the subterranean minerals and water. And the animal facilitates the geographic dispersal and the continuous recycling of the organic matter.
It's a massive, continent-spanning mutual survival pact. The bacteria process the chemical waste, the fungi transport the goods, the invertebrates maintain the physical environment, and the vertebrates manage the geography.
And understanding these deeply complex, interconnected relationships is the foundational scientific argument for organic agricultural practices.
Right. If you view the soil merely as an inert sponge holding up a plant, dumping synthetic chemicals into it seems logical.
But if you understand that plant health relies entirely on this delicate living metropolis, the calculus completely changes. The primary concern is that broad-spectrum pesticides or highly concentrated synthetic salts might damage or short-circuit these intricate natural cycling mechanisms.
Exactly. If a chemical application inadvertently wipes out the aerobic bacteria, the nitrogen refinery shuts down.
If a fungicide collapses the mycorrhizal network, the plants lose their access to deep water and phosphorus.
If agricultural practices kill the earthworms, the pH buffering ceases, the soil compacts, and the whole system suffocates. Every single job in the metropolis matters.
It is an economy of survival where absolutely nothing is wasted, and every organism's waste product is another organism's vital resource.
Let's summarize the journey we've taken today, because the sheer scale of the biology we've discussed is hard to hold in your head all at once.
It's a lot to take in.
We started at the absolute smallest scale, looking at a single teaspoon of dirt containing up to 3 billion bacteria. We explored how autotrophs, diazotrophs, and anaerobes run the complex chemical refineries of the nitrogen cycle.
Pulling fertilizer directly out of the atmosphere and refining organic waste.
We then followed the nutrients along the fungal highways, visualizing the 700 kilometers of microscopic mycelial threads woven into a single gram of soil.
We saw how these threads form common mycorrhizal networks, linking individual trees into a vast, nutrient-sharing superorganism capable of subsidizing its youngest members.
We climbed the size scale to watch the invertebrate engineers at work. We looked at earthworms acting as mobile composters and pH buffers, constantly neutralizing the soil chemistry so both fungi and bacteria can thrive.
We saw ants and termites manually mixing the deep mineral soils with the organic surface layers.
And finally, we examined the heavy machinery, the vertebrate mammals engineering the landscape to capture water and prevent erosion, and acting as biological couriers to spread the fungal spores across the continents.
The ultimate takeaway here for you, listening to this, is a complete paradigm shift regarding the world outside your window.
The growth of a towering forest or the vegetables in your garden is not just a simple equation of a seed getting some sunlight and rain.
It is entirely 100% dependent on the continuous, frantic, unseen labor of millions of specialized organisms beneath your feet, constantly breaking down death to manufacture the building blocks of new life.
It demands a deeper respect for the ground we walk upon. It is not an inert surface. It is a highly active, living entity.
And as we wrap up, I want to return to that final provocative thought you promised earlier.
Oh, right. During our discussion on fungi, we talked about a specific protein called glomalin.
The biological glue produced by arbuscular mycorrhizal fungi.
Right. This microscopic glycoprotein physically binds our global soil together. It keeps the earth from blowing away as dust. It creates the spongy texture that allows water to penetrate the ground.
And it stores a massive, unbelievable portion of the Earth's entire supply of terrestrial carbon and nitrogen.
It is a fundamental, load-bearing pillar of the global ecosystem. Our entire worldwide agricultural food supply ultimately rests upon the physical structural integrity provided by this specific protein.
And the staggering detail from the scientific literature regarding its discovery.
Glomalin was only discovered by scientists in 1996.
Wow.
Think about that timeline. 1996. We had already sent robotic rovers to the surface of Mars. We had split the atom. We were actively mapping the human genome. We had built a global internet.
But we had absolutely no idea what was physically holding the dirt in our own backyards together.
We were entirely blind to one of the most critical biological mechanisms on the planet.
That is just wild to think about.
So considering that our entire world rests on this delicate, fiercely complex subterranean economy, I want you to mull over this as you go about your day. What other massive, world-sustaining mechanisms are operating right beneath our shoes at this very moment, completely invisible and totally unknown to us?
The soil metropolis undoubtedly has more secrets to share.
Keep digging. We will catch you on the next Deep Dive.